thank you for having us until now in this panel. The next talk is for Dr. Mehmet Dokmeci, Associate Professor of Terasaki Institute for Biomedical Innovation, with talk title of Organ on Chip or Microfluid Systems for Modeling of Wound Healing and Angiogenesis. Dear doctor, hello, how is it going?
Good, thank you.
We are really happy to we are really happy to see you in this meeting. Thank you for having us in this session. We are ready to listen to your presentation. Please start your presentation.
All right. Start sharing. Do you see my screen? Do you see my screen?
Yeah.
All right. Good morning, everyone. First of all, I want to thank the organizer for inviting me to this conference. Appreciate it. I work at the Terasaki Institute for Biomedical Innovation with Dr. Ali Kadem-Husseini. First of all, let me give you a little bit introduction about our institute. The first organ transplant shown on the left side was at Brigham and Women's Hospital between twin brothers. It was a kidney transplant. Starting with that, there was a major development momentum in organ transplants. Dr. Paul Terasaki on the right side developed a cytotoxicity kit that develops cells from the donors with blood from the recipient to understand whether there's a mismatching problem. He developed the technology. He had a very successful company called One Lambda and then started the institute. Initially, the institute focused on testing solutions for organ transplants, but later on, it transformed into other areas. Starting from last July, Dr. Kadem-Husseini and I moved to the institute, and these are the areas we are working at. We're working on biomaterials, implanted devices, organ on a chip models, and personalized devices. And how do we do it? We bring an interdisciplinary group of engineers, clinicians, and chemists to come up with novel solutions to current problems. And we also have a business development team as well as a tech transfer team. And at the end, we either start up companies or train the future thought leaders in terms of postdocs. Let me go back to the theme of the conference. It's about wound healing, and we developed a smart wound healing patch. As you know, the chronic wounds occurring in diabetic patients take multiple months to heal. During this time, if an infection occurs, it goes unnoticed and may result in major problems, including amputation. And our idea was to create a smart patch system with embedded sensors on the side. It's a two chip system. One chip is the right part where you have sensors for temperature, oxygen, pH that monitor the wound in a real time, and an electronic component that senses, that processes these signals, either sends it to the cloud, to a clinician or a healthcare unit. In addition, if there's a minor infection or some type of infection, can trigger the microheaters on the substrate to deliver antibiotics and drugs for rapid intervention. This is very important, especially for rural areas with limited access to medical care. We all know that the pH and temperature variations are a sign of infection, not alone, but in a combined unit. And if you can release the antibiotics in a timely manner, you can prevent major problems with the infection. We do not want to release antibiotics continuously. Hence, we developed a microheater system that can deliver these drugs in a very controlled, on-demand, when needed manner. Here is a big picture of the device we've developed. We have included pH, oxygen, temperature, and strain sensors. Also, it's a smart and self-powered wound dressing. It can deliver drugs, can also release oxygen in a closed loop manner, meaning we sense the environment and only deliver drugs or oxygen when needed, when the infection occurs or the oxygen levels are less. We have also tested these in an in vivo skin model that I'll show in a few slides. We've gotten quite a bit of press with our technology by NSF, BBC, and CBC, and you can look at these YouTube videos if you like. Here's an overview of our devices. We built oxygen and pH sensors that I'll explain in detail later. We've built microheaters that can release drugs on demand with signals from electronics, and we also built this closed loop 2-unit system, a disposable sensor patch which can be replaced every few days, as well as an electronic unit that is designed to obtain the signals, process them, send it to a remote hospital, as well as trigger signals for drug release. Here is the overview of our stimuli-responsive drug delivery platform. We are using poly-NIPAM, temperature-sensitive drug delivery system. We put drugs into microparticles and place them into gels, place this big system into the gel, put it on top of a microheater. When the microheater receives the signal, heats up the gel, the microparticles shrink and then release the drugs. All done and controlled by a chip placed nearby the microheater. Here's the process. These are poly-NIPAM particles at 25 C. The dimensions are about 600-700 microns. When you increase the temperature to 37 C, they get smaller and release the drugs. As you see that the dark spots, when they're smaller, the drug is being released. The data below shows that at 25 C, we don't have much release, but if you increase the temperature to even 37 C, there's much of the drug is being released. This is 50 conjugated dextran. You notice that 37 C is not in direct contact with skin, so it doesn't cause problems. It's on top of the hydrogel is the one that's touching the skin, and the heater is touching the hydrogel. So the direct contact of skin is not reached within the system. Here's the drug-embedded gel, our microfabricated microheaters, and the microheaters with the drug-embedded gel shown on the skin. Next, we have created polymeric nanoparticles with using electrospun fibers. With the electrospun fiber sheet, we have created flexible and conformal substrates. These are biocompatible substrates, compatible with any liquid environment, and easy to fabricate and very flexible. We also placed a microheater on this nanofiber substrate, as well as an antenna for receiving the signal. Before you apply the heat, the drugs are encapsulated in the nanofibers. When you heat it up to a 35 centigrade, you can release the drugs. This is the system with embedded drugs inside the nanofibrous substrates. Here's our nanocarriers made about 50 to 60 nanometers. Both cefazolin and ceftriaxone, which are antibiotics, are loaded into the nanoparticles. And when you increase the temperature, we've noticed that you can release methylene blue at different temperatures. Red one is 25 C. If you increase the temperature, you can notice that the cumulative release increases over time. We've done a continuous release as well as a cyclic release. Right? That means we increase the temperature, then we stop. When it stops, the curve is flat. Increase the temperature again, the curve is flat. Increase temperature, the release is increased. And when you stop the temperature, the release stops. So we have a good control about the amount of drugs that are being released over time. Here's we talk about closed loop drug delivery. That is, we sense pH, we sense temperature, and when needed, we trigger the heaters to release the drugs. We've also worked on biological closed loop. This is a project that we're very proud of. We do not want to release drugs continuously. That's not good for the cells. We want to release the drugs when needed. So if you look at the wound biology, some inflammation is necessary for healing. However, excessive inflammation can impede healing. Wouldn't it be nice to stop inflammation when there is overinflammation? And then we use the ibuprofen drug with a smart system that would release the drug when there is a lot of inflammation. In a nutshell, the device is shown in the middle. We have polystyrene microbeads that are the carriers. They have a cleavable peptide with amine groups, and the drug is attached to the end. When there is a inflammation, this one cleaves the peptide and the drug is released. When there's no inflammation, the drug stays where it is. Hence, this is a biologically closed-loop smart system. Here is the test results that shows that we're showing the pro-inflammatory macrophage activity is being suppressed with ibuprofen, as well as when the correct sequence, when the inflammation activity cleaves the peptide, and hence the drug is released, shown here. When you use a different peptide sequence, when you just use regular beads or control, you don't see any suppression of the inflammation activity. We've also used freezer paper to make flexible devices. Here, we have a finger actuatable pump that can deliver drugs from a chamber using the wicking activity into the wound area. We were able to dispense multiple volumes over time in a channel of 150 to 500 microns. We can release low viscosity liquids as well as high viscosities with a pump that can operate at a flow rate of 0.4 microliter per minute. We've also used the freezer paper. This is standard commercially available low-cost paper to create an air-liquid interface for culturing epithelial cells. The system is shown on the left top. We have a media inlet that goes to the media transport layer using wicking action, and the cells are encapsulated inside the hydrogel matrix. One side is the air, the other side is the liquid, similar to creating a skin-on-a-chip type of application. We noticed that when you have flow as expected, the cells are alive. Under static conditions, the viability of the cells go down. But the system worked quite well, and the next step is to create skin-on-a-chip type of devices. We've also made a flexible pH sensor on a transparent membrane. You can see an array of sensors. The sensors have a linear response. They work at least 24 hours. No leakage current no crosstalk between different devices, and they're quite fast, about 4 minutes. We are measuring the open circuit potential with a linear response in terms of pH. We've also measured the sensor with different pH values. As you notice, you can obtain different staircase values, which is very stable and with reproducible results. We've also generated oxygen. How do we do that? We create this PDMS platform. The light blue is the peroxide inlet. It interacts with the cobalt catalyst. Cobalt catalyst releases oxygen to the wound area on demand. Peroxide is not good with cells, hence it's isolated from the wound area with a parchment paper and a PDMS layer. There's a cross-section fabrication of the device using microfluidics. The same device, which is fabricated on a parchment paper. We've also created oxygen sensors and integrated on the same device. The flexible devices paper is not very compatible with skin. It's not that flexible. So we created a hydrogel elastomeric interface layer in between our devices to the skin. These parameters are flexible. They preserve drug activity, breathable, and can be easily microfabricated. One of them, alginate, is very fragile, and glycerol, we wanted to add the mechanical properties, so we mixed them together. If you notice, that we can have enhanced Young's modulus with these systems. After 72 hours, they're still flexible, and they are, even in the dried form, they can absorb large strains. We looked into the exudate uptake and obtained good results. We've also created an oxygen sensor. It's a current-based oxygen sensor with a linear response in terms of the oxygen concentration. It is sensitivity as 0.5 microamps per percent oxygen. And the inset, the time response is about 20 seconds, and the output current is proportional to the amount of oxygen, dissolved oxygen in the media. We've also connected to an antenna to make a wireless system. We've also delivered drugs into bacteria cultured in a petri dish. The bacteria used was Staphylococcus aureus. And we noticed when the bacterial culture changes the pH of the media by 0.4, the drug release is activated, and the dark area, as shown here, is the part where the bacteria has died. Hence, we were able to activate the drug release and kill the bacteria on demand. Here's our full system. The flexible heaters are on the side, and it's a wearable unit with the having drug release data shown in the middle. Next, we also developed the in vitro skin model to test some of our devices. In our lab, we use a lot of gelatin-based materials, including gelatin metacrylate. That is gelatin metacrylated with additions. It's photo cross-linkable. We had a photo initiator. We cross-link the hydrogel. Then we seed the keratinocytes, and we bring them to air-liquid interface to create artificial skin after 7 weeks. We have initial good results. You see our gelatin metacrylate platform after 2 weeks. After 6 weeks, we generated stratified layers. This is compared to a gold standard of collagen. After 6 weeks, our results are quite similar, histology results, and this is the human skin. Again, the epidermis thickness between JALMA and collagen, they track each other quite well. Here's confocal images that shows that the cells are active and highly placed in the JALMA scaffold, which is quite comparable to results achieved on collagen, and here is and also compared to the human skin. Let me tell you about latest results. We're very interested in microneedles. We use microneedles for drug delivery, cell delivery, diagnostics, as well as gene delivery. These are cell delivery. We place cells, including mesenchymal stem cells, into JALMA. JALMA is quite soft, which allows the cells to be alive, viable, and metabolically active. However, the softness doesn't allow the JALMA needles to penetrate the skin. As such, we place a PLGA shell around these microneedles and place them on a flexible detachable substrate. So we press the substrate. We take the patch, insert it into the skin, which it can due to the hardness of the PLGA shell. And then we remove the substrate. Over time, the PLGA shell degrades and the cells are released in a very quick manner. We've also, these are the confocal images that shows that the PLGA shell, is penetrating the skin. We were able to deliver 3T3s as well as MSCs using this approach. These are shown in vitro results, and this is the microneedle tissue insertion into cardiac muscle as well as skin. And these are the animal results that show that the cells can be delivered and enhance regeneration. We've also used these microneedles for extracting interstitial fluid. The way it works is we create a PDMS mold, pour the JALMA and the photo initiator inside the mold. We cross-link using UV. We we dry and demold, as well as remove the microneedle patch from the mold, and then apply the patch to the animal. The JELMA patch extracts the fluid from the interstitial fluid from the animal. You centrifuge and remove the microneedle, and you have the sample for testing. We were able to apply these devices to small animal models, and we were able to extract different interstitial fluid. And within that, we were able to measure glucose concentration, which is similar to the glucose concentration from blood, as well as vancomycin concentrations. We were able to also deliver anti-cancer drugs, including doxorubicin. We take gelatin, we metacrillate it at 50 C. We attach doxorubicin, and then we place this pre-polymer solution into a PDMS mold. We cross-link, and then we demold it. We split the needle from the PDMS mold, attach it to the skin. After swelling, the drug release happens, and then the doxorubicin locally delivered. This is mainly for melanoma type of skin cancer applications. These are the sustained drug release results that we obtained using our patch. We were also able to deliver curcumin, another anti-cancer drug. We take our gelatin base, we do a BCD conjugation, which allows us to attach curcumin onto these sites. We follow the same process using a PDMS mold, create the microneedles, and attach this on a 3D tumor models to deliver curcumin. Here is curcumin-loaded microneedle patches. They are very uniform, fabricated in a high-rate manner. They're inserted to the tissue, and you can see the sites where the microneedle has been inserted, and we were able to deliver different amounts of curcumin into the tissue and killing the cells. We were also able to use gene delivery by embedding the DNA with the PBA inside the microgel, inside the JELMA microneedle, and then by insertion can deliver genes as well and transfect the fibroblasts. We were able to demonstrate these in a 3D in vitro model, as well as efficiently deliver the genes in an in vivo model. We were able to use microneedles to deliver MSCs in a regenerative therapy, as well as tissue regeneration. Here is our team, and I'm ready to take any questions.
Thank you for your comprehensive talk and great presentation, Doctor. May I ask a question? What are your challenges for treatment of large skin defects? I mean, could you scale up this methodology, this product for treatment of large skin defects or not?
At the moment, we're the technology we're using is...
turn off your share screen?
I turn off the share screen.
Thank you.
Our technology is currently used for a couple centimeters, but the technology is microfabricated, so we can go up to, it's on flexible substrate, which depends on the mold. We can go up to tens of centimeters if needed. The good part of this microneedle technology is flexible and conformal. So if the wounds, the wounds do not come one size, it comes in different sizes, different depths, different tissue parts. We can, using this flexible conformal platform, and apply it to any type of wound that is needed.
Thank you so much again. All the best, and I wish the best for you. Be safe and goodbye, doctor.
Thank you. Take care.